Okay, good afternoon, everybody. Welcome to today's Coffee Science Seminar. Can we have the next slide, please? So today's seminar will be given by Professor Eugeni Roura, a professorial research fellow in coffee, with an intriguing title of 3 weeks to nourish the chicken embryo. Next slide, please. My name is Mike Gidley. I'm Emeritus Professor in coffee, and it's my privilege to chair this session and to introduce Eugeni and to facilitate the Q&A session at the end. Next slide, please. So at the University of Queensland, we acknowledge the traditional owners and their custodianship of the lands on which we meet and pay our respects to elders past and present, and acknowledge the unbroken link with country over 65,000 years. So this seminar is scheduled to finish at 1 p.m. And for any matters arising, please type your questions into the Q&A tab. That is the only place I'll be looking for questions. So unless you put them in the Q&A tab, I won't see them and Eugeni won't get to answer them. So next slide, please. So it's my pleasure to introduce Professor Eugeni Roura. Eugeni is a veterinary nutritionist by background with broad research interests in digestive physiology, chemosensory science, that means the science of taste and smell, as well as transgenerational nutrition across monogastric animals, so chickens, pigs, and humans. Today, we're going to hear about his work in chickens. After a significant professional career in industry in Europe, Eugeni joined UQ in 2010, where he leads a large team that's active at the interface between basic and translational research. And his aims are for large societal applications across agriculture, food, and nutrition. He's graduated a large number of students and published a large number of papers, but also very active in the community. So he's the president of the World Poultry Science Association Australia branch. He's a member of the National Committee for Nutrition of the Australian Academy of Sciences and director of the AgriFutures Chicken-Meat Consortium that we'll hear about some of the work of today. And I'm also pleased to have this opportunity to congratulate Eugeni on the recent award of the Senior Medal by the Nutrition Society of Australia that recognises excellence over time in the science of nutrition. Well done, Eugeni. And today's title is, next slide, please. 3 weeks to nourish the chicken embryo. And I think now the floor is yours, Eugeni. So remember, everyone, put your questions in the Q&A tab, and I'll come back and host those questions at the end. All yours, Eugeni.
Thank you so much, Mike. I appreciate your kind words. And thank you to the organizers of the coffee seminar, particularly Maximiliano Müller, for inviting me to share some of our research that we've been working on over the last few years. Now, I too would like to acknowledge the traditional custodians of the land where we meet, the Turrbal and Jagira people, the elders past, present, and future. Now, let me just start with this, as Mark said, intriguing title, 3 weeks. And 3 weeks is what it takes for an embryo to develop. This is, by the way, how pretty much looks a chicken embryo of 7 days of age. My point will start with the egg, of course. What else if not? This is an egg. And probably it inspires different things to all the attendees to this presentation. And thank you, by the way, for listening to us. From me, an egg, I think about an egg from a nutrition point of view. It's the most balanced diet that you can ever think of. It has perfectly balanced amount of energy to protein, essential amino acids, non-essential amino acids at the right proportion. It has vitamins, it has minerals, everything, in fact, everything that you need to generate life. And that's the other point of view of an egg. For me, an egg, especially a fertile egg, is the miracle of life itself, right? Because out of a fertile egg, we start with the yolk, and 20, 21 days later, we have a being, an independent being, a walking being, a being that will be eating, that will be screaming and shouting and smelling and tasting and everything that goes with it. So that's pretty much sort of a miracle, isn't it? Such a small round object that will end up with broiler chicken. So it's fascinating to try to understand this process. How do we go from an egg being laid and 21 days later having a day-old chick? There's several constraints during that space. And if you follow my cartoon, there's a number of aspects that I wanted to touch that will introduce the topic. One, obviously, is that the chicken that we know, the broiler chicken, grows very fast, and genetic selection has taken that chicken to be growing in 5-6 weeks. We have a chicken that goes from 40 grams a day old to 4 kilos, right? The rate of growth has been evolving as it does genetic selection. And so nowadays, as I said, you don't need much more than 40 days to have a fully developed broiler chicken. When we looked at the embryonic development, there's a bit of a different picture. 50 years ago, it took us 21 days for the embryo to hatch. And today, it takes 21 days for an embryo to hatch. Isn't it interesting when the genetic potential of the animal has evolved so much? So what's going on? Is the genetic potential present in the embryo? Of course it is. Why is not the embryo growing faster and bigger as it does post-hatch? That's a few good questions, and some of the aspects, maybe we'll touch on them. But what's actually crucial is that at hatch, that day-old chick is actually not fully developed yet. So that supposes a challenges. I have named this slide as the big transition, because the embryo will go from basically feeding itself from the different egg compartments, liquid, yolk, albumen, and that will obviously be the source of all the nutrients that the embryo needs. Once it hatches in the post-hatch, all right, the chick all of the sudden has to find a way and will start eating solid foods, etc. So big transition, very challenging, especially if we consider that the embryo is the one-day-old chick is not fully developed. Now, another aspect of this slide, and I'll change soon, but I will go back to the oviposition day, so I'm going backwards. So now we have 6 days somehow through the broiler breeder to potentially influence these developing embryo, right? So all in all, we have 6, and then the 3 weeks that I was mentioning in the title of my speech. And that's it. Once we finish these 21 days, then the chick is on its own, and then it starts the more classic. Now, be aware that when we talk about the one-day-old chick, that lack of full development, particularly of the gastrointestinal tract, but also the immune system, etc., causes a number of problems that we're trying to solve. And those problems will be related to the fact that the chicken is not able to digest all the solid food that needs. Maybe it's not fully prepared for the challenges of the outside world. And therefore, that improvement in the maturation of the digestive system and the immune system is fundamental, and it's a true challenge. In the first days post-hatch, it's then that we see quite a high mortality rates that may go from 2 to all the way 5%, and some of the survivals will still be lagging behind the rest of the flock for the rest of their lives. So this is truly the crucial step in the life of a broiler chicken. Now, my talk will be divided into 4 blocks, or if you want, in 2 blocks, And within each block, I have 2 themes that I want to talk about. Now, the first block is around how can we influence the embryonic development through the parental generation, broiler breeder nutrition, and how by influencing broiler breeder nutrition, we can have an impact on the progeny. We call that intergenerational conditioning, or transgenerational if it includes more than one generation. And I'll be talking about chemosensory mechanisms and epigenetic mechanisms. Now, the second half of my presentation will be around the directly interventions in ovo. So can we supplement the development of the embryo so that we improve that development as much as possible? And we have come up with a couple of very interesting topics that I will be talking about. One is about acidifying the environment, the in ovo environment, and the other one is about novel applications of functional compounds, particularly essential oils. So bear with me, I'm going to start talking about broiler breeder nutrition and chemosensory and epigenetic mechanisms. That's, again, what it takes for an egg to be laid. It takes usually around 5 days for the ovary to produce the oocyte, and then it gets fertile after these 5 days. We have 1 more day, around 24 hours, for the final formation of the other components of the egg until it's actually being laid. All in all, it's 5 + 1, 6 days. So we have 6 days that the egg is inside the breeder that will be impacted by factors that are influencing the breeder themselves, right? And that's what I'm going to be talking about. Some of these aspects will be based on sensory perception, and that's my topic of the next 4 or 5 slides. It's a fascinating story, honestly, and I'm biased, of course, but what happens is that when we talk about the external environment, we all perceive a number of smells, if we start eating, taste, textures, etc. And these perceptions may have an impact on a number of physiological parameters in ourselves. But that also is true when it comes to the broiler breeders, and while they are laying the eggs, some of those sensory stimulus will be transferred and will be impacting the egg. More precisely, I'm talking about volatile compounds coming from the external environment, and more precisely, the ones that we actually controlled relatively well are plant volatile compounds. Now, when we're feeding a broiler breeder, we're feeding them cereals and protein sources such as soybean, etc. And these plant cells are actually producing volatile compounds that eventually might be affecting the developing. Now, when it comes to chemosensory mechanisms, when it comes to the production of plant volatile compounds, there's nothing random. And we've learned over the years, particularly the last 10, 15 years, that the group, if you want, the conjunction, the total amount of plant volatile compounds of a given cell, we call that the volatilome. It's the group of all the volatile compounds produced by a plant cell. This group of volatile compounds, they're actually quite costly to synthesize. And most of the times, the precursor of these volatile compounds, as it is depicted in the slide, come from actual nutrients that otherwise the plant could be used for other purposes. But the plant decides to use some of these nutrients, such as fatty acids, amino acids, vitamins, minerals, not minerals. They decide to use that to metabolize those, synthesizing what we call the volatilome, the plant volatile compounds. Those volatile compounds are not necessarily immediately released. In many cases, they are stored in vacuoles and only released when it's the appropriate time. I'm thinking of a classic example is the tomato or the strawberry or so many fruits. that these volatile compounds have been stored for probably days, weeks, maybe months, and until the fruit is ripened, then they are released to attract consumers, if you want, predators, and then the seeds are dispersed and so on. There's an evolutionary advantage in doing so. But my point is that those volatile compounds are actually nothing. It's random what happens with those volatile compounds. Now, what is that to do with my 3-week story? Sorry for going all the way around. I would like to acknowledge the contribution of Dr. Marta Navarro and others in this topic, like Sally Taylor. What we've been doing in our group is characterizing the volatilomes of all the main feed ingredients that the broiler breeder, or that also applies to pigs, will be fed. In the left side of the screen, you will see a relatively standard broiler breeder ration, a broiler breeder feed formulation. And as you would expect, without needing to be a nutritionist, the main ingredients would be cereals and soybean as main protein source, some oils, and then, of course, all the supplements for essential amino acids, vitamins, minerals, etc. Now, 80–90% of this formula is these 3 or 4 ingredients at the top: wheat, barley, sorghum, soybean, etc. So let's look at the volatile compounds that these ingredients may release. We have characterized around 34 or so main feed ingredients used in poultry diets, and we can identify roughly around 600 to 700 individual volatile compounds present or released at some point by these 34 main feed ingredients. Now, in the slide, you will see 8 or 10, the most abundant in wheat, in barley, in sorghum, and in soybean meal, right? Some of them are repeated. Hexanol is present in most, is quite abundant, but some others would be relatively unique and very typical of specifically that source. Now, what happens then is that these feed volatiles are then consumed by the broiler breeder. That's what we feed them. We feed them a balanced diet. And while what we do is looking for the nutrients of those ingredients, inevitably, they will also be exposed to these volatile compounds. And what happens with the volatile compounds? Well, as you see, then we identified a number of these volatile compounds. And from the feed, they will be transferred into the plasma, not at the same rate. Some of the volatile compounds are easier transferred than others. Some volatile compounds will be more than others. But in general, a number of them, I'm going to take as an example, caproic acid is at a reasonably high abundance in the feed and is the most abundant that we found in plasma. But also, because the broiler breeder is actually producing fertile eggs, those volatile compounds from the plasma then will be inevitably transferred to the developing egg, fertile egg, right? And as you see, those volatile compounds can be traced back all the way to day 14 of embryonic development and to the end of the embryonic development, clearly telling us a story that the developing embryo during the 3 weeks will be constantly exposed to these volatile compounds coming from mom's diet. It will be swollen, it will be ingested, and eventually they will be stimulating the sense of taste and smell in the embryo. How important is that? Well, have a look at this graph. I hope you see the graph now, and I would like to acknowledge the master student, Rafika Tunisa. She did a great job in this experiment. Now, what we did in here is give the first week of the life, post-hatch life of a chicken, give them a choice between 2 diets. And those diets were diet one, formulated, mimicking, if you want, the standard diet of the broiler breeder. So it's a high wheat and soybean diet. While diet 2, yellow, it's a completely different diet. No ingredients that mom broiler breeder had been exposed to. It's a diet based on corn and a protein hydrolysate, both very highly digestible. If you ask me from a nutritional point of view, diet 2 is actually probably better, much more expensive than diet 1. But if you ask the day-old chick, see what happens, and that's the graph to your right. It just expresses the preference for diet 1, 75%, compared to diet 2, only 25%. So let me just explain this, because I've been doing research, behavioral research with animals all my life. And those 4 asterisks here relate to p-values. These are so highly significant differences, p less than 0.0001. Those of you who might be familiar with animal science and behavioral studies, it's unseen. This is the day 1, so 12 hours after hatching. The chicken already knows what to eat, and they go for mom's diet. Interesting. Very, 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 very impressive. Of course, we know that happens also in humans, and probably it happens in pigs as well. We're doing some experiments in that way. So there is a tremendous impact of maternal diets in what the progeny will be choosing, and that is a fantastic tool to take into account for future research as well. Gears now, and I'm going to be talking about, still with the broiler breeder, but I'm going to tell you a little bit more about all the mechanisms. So I've talked about one example on the chemosensory mechanisms. I'm going to talk to you about potential epigenetic mechanisms. I'm going to be cautious in the sense that I don't actually have epigenetic data to present you, but what we're presenting to you is proteomic data. So we're comparing 2 different treatments, as I will explain next, but it's proteomic data. And the reason I'm talking about epigenetic mechanisms will be explained later on. But I would like to acknowledge some of our contributors. Number 1, the PhD student who did actually the experiment with the broiler breeders, Bipul. I also want to acknowledge our proteomics expert, Xinle, and our collaborators from the Center for Animal Science, Shilaume and Lon Nguyen. Thank you all for your contribution. But the main student involved in this experiment was the honors student, Norman Lai, who did a fantastic job. So the experimental design is relatively simple. We wanted to know if the broiler breeder diet will have an impact on the progeny adapting to a harsh environment, for example, a low crude protein diet. So there you go. That's and I'm going to the wrong eye. So basically, we were asking whether broiler breeders fed a 16% crude protein diet, which is a relatively high crude protein diet for a broiler breeder, compared to broiler breeders fed at 12, a low crude protein diet of 12%, would have progenies that would deal differently with their own levels of crude protein in the diet. I'm not going to explain today because it's not, I don't have enough time. I'm not going to tell you the results in the progeny, which basically were not that successful. The progeny did not adapt well to low crude protein diet, even if mom had a low crude protein diet. But that's a different story for another day. Maybe Bipul will be able to explain you much better than me about this. But what I wanted to talk today is about mechanisms that are transgenerational or intergenerational. And those mechanisms point at epigenetic mechanisms. So we were comparing chicks born from breeders with a low crude protein diet to the jejunum of chickens born from low crude protein diet out of 12 compared to 16, compared to the high crude protein diet, 16%. Now, long story short, and I know I'm gonna jump here quite quick, but because my whole point is that we are uncovering a novel field of study, at least, of course, in broiler chickens, that will try to link the diet of the broiler breeders to the resilience and the development of the developing embryo and the old chicken. And it will use mainly epigenetic mechanisms as it is shown by these results. And I'm probably going to jump over this slide to directly go to what proteins were actually found as the most significant when comparing these two groups. And remember, I'm comparing chicks born from breeders of high protein diets to chicks born from breeders with low protein diets. And the modification of the proteins pointed at histones that, as it is well known, the methylation of histones is one of the main mechanisms of epigenetic of the epigenome. So future experiments, of course, will be knocking back at the door of Sheila O'May and Lone to help us discern and to better understand those epigenetic mechanisms that have an impact from the broiler breeder to the progeny. Now, of course, those proteins would be having an impact on a number of pathways, and I'm not going to be describing them in detail. But as you would imagine, when we talk about the low crude protein diet, it means that that has an impact on how carbohydrates and amino acids are being used metabolically. So you change the diet, you change the metabolism of the eukaryotic cells, and the main impacts were in glycolysis, glyconeogenesis, pyruvate, therefore the tricarboxylic acid, amino acid metabolism, tryptophan, fatty acid metabolism. The way through, and I do have another 12 or so slides that I would like to talk to you. The first part of my talk has been about transgeneration, intergeneration. The second half of my talk starts now, and I'll be focusing in direct interventions in ovo. That is, while the embryo is being developed. And in our group, we have two main topics that I want to talk about. We've done also through Mila Meyer, PhD, we've done quite a lot of work also on the immune system, but I don't have time, so I had to select what to talk about. And I'm going to be talking about metabolic changes first, and particularly acidification. And then I'll be talking about application of functional compounds, essential oils, and how those may have an impact on digestive development in the developing embryo. Let's start with the metabolic acidification. This is a topic really that we stumbled into. We were not initially thinking of working on pH, but the idea was working with a number of essential oils. And as you know, many of the essential oils have quite an acid pH. So my student Asad Ali and myself, we were a bit concerned about how the pH would have an impact on the embryonic development. And to start with, we started looking at what was the pH of the different compartments in the egg, and there was not much information, etc. But ideally, what we did come up with is that the observation that the production of CO2 during embryonic development have a tremendous impact on the pH. That was not necessarily what we were looking for, but it turned out to be a fantastic story that I want to share with you. Now, what happens with the production of CO2 is that in initial stages, when the egg is being formed inside the breeder, well, the partial pressure of CO2, we call that PCO2, will be in equilibrium with the hen tissues, right? The plasma of the hen, etc., etc. So I call that isobaric period, right? Once the egg is laid and in the initial stages of incubation, there's not much going on. There's very few cells in the germlines that are being reproduced. So the amount of CO2 produced is very low because it's directly related to cellular metabolism. And therefore, during that early stages of embryonic development, there's a net loss of CO2 through the pores of the eggshell. I call that the hypobaric phase. And then later in the stages of embryonic development, the closer we get to hatch, the more cells are reproducing, metabolism increases, and therefore the production of CO2 massively increases to the point that the amount of CO2 released by the pores is insufficient, and the pressure, the partial pressure of CO2 increases to create an environment that I call hyperbaric to partial pressures of CO2. Now, just as a refresher, where coming from. And as I said, it's all about cellular respiration, metabolism, right? And if we go back to our Biochem 101, if you want, and I assure you that I had to go back to review the concepts, but they are really fairly simple. It's all about producing energy, producing that energy that can be used by eukaryotic cells. This is ATP. And in the production of ATP, we're using skeletons from carbohydrates or amino acids that will eventually enter the mitochondria, the Krebs cycle, the tricarboxylic acid cycle, and eventually at our spinning of the cycle, we will be producing some ATP. But with the ATP, as you probably now may recall from your Biochem 101, there's also the production of CO2 and water. So therefore, that's what CO2 is coming from. So for every cycle of glucose, you will get 6 molecules of CO2 and a number of molecules, 30 or so, of ATP, which is what actually the cell needs. So let's have a look at the impact of the production of this CO2 in the internal environment of the developing embryo. Well, as you see in this work published by Branham and co-workers in 2022, clearly in late stages of the incubation, say days 12 to 19, remember that the hatching occurs on day 20-21. So late stages, you see actually this accumulation of CO2, which in turn gives us that increase in partial pressure of CO2 as the partial pressure of O2 decreases. Now, this partial pressure of CO2 is associated with, and I'm now looking at the graph to the right, it's associated with a decrease in pH. As you see, when we go from day 12, 14, 18 of embryonic development, the pH of the blood goes from 7.65 all the way down to 7.5, 7.48, right? So there is a clearly trend of acidifying, and that's the bicarbonate buffer system. CO2 plus water will give us bicarbonate, and then if we further push the system, HCO3 negative and H positive, so hydrogen ions will be produced. And when we produce a massive amount of hydrogen ions, the pH decreases, and therefore we're acidifying the environment. So we were wondering then what happens in these different compartments that are responsible for the development of the embryo. And the main compartments are the albumen and the yolk. These are the major compartments, although we do have some other compartments that I'm not going to be talking about because they are minor and they disappear in 3 or 4 days. So yolk and albumen, as you see, we looked at the buffering capacity of those compartments. And the gray line is just simple water. As soon as we add some citric acid, the pH decreases sharply, and that's it. Now, PBS is a commonly used buffer system in our labs. And yeah, it does buffer quite well. After 1.5 mils of citric acid, but then starts decreasing when we go above 2 and 2.5 mils. Now look at the red line, the yolk. The yolk is impressively stable when it comes to pH. So it takes a lot of hydrogen ions to modify that pH. A similar story can be told about albumin. So both the two main egg compartments have a brutal capacity, buffering capacity. And it makes sense. It makes sense in the context of what I'm going to be explaining you next. And that's, we looked at the potential impact of that hyperbaric phase of late embryonic development and how changing the pH could have an impact on the embryonic development. And that's my student, PhD student, Asad Ali, who is now finishing his PhD, who worked on that project. Well, the first is the characterization of the pH dynamics in albumen and in yolk. And as you see, parallel to what you would expect in the production of CO2, the higher production of CO2, the higher the production of hydrogen ions, and the lower the pH of albumen. Now, what happened there is that at the initial of the incubation, the albumen is pretty alkaline. So it does get a quick descent of the pH as we get closer to hatching. Now, when we look at the yolk, it's not the same. The yolk, it actually, during the first stage of embryonic development, increases the pH, and it's only at late stages, probably because the production of CO2 is massive, that this pH start declining. What are the physiological meanings of these findings? All right, so here in the pH upper right corner graph, I'm showing you the different compartments, amnion and allantoids, which we haven't talked about, but the main ones, albumen and yolk, and how their pH changes over time from day 0 to day 20 of incubation. Now, this range of pH, somewhere between 6.5 to 8.1 or 8.2, is in fact coincidentally some of the optimal activity of the enzymes in the yolk sac, such as lipase. So these are optimal pHs for the activity of the hydrolytic enzymes of fats. Now, when we looked at hydrolytic enzymes for protein, aminopeptidases and so on, we're far from the optimal. But as we get closer to hatching, the pH of, say, albumen keeps going down, also of the yolk, and therefore, the closer we get to hatching, the higher the activity of the aminopeptidase, because as you see in the scale to the left, relative activity, the relative activity increases as the pH decreases. So there is a potential that by playing with the pH of the different compartments, we could change the activity of the enzymes that will be hydrolyzing proteins and fats and will then release nutrients for embryonic development. So final pH of yolk at hatch and albumen, when it finally disappears, it's 6.88 for yolk, 7.18 for albumen. And I'm saying these numbers because that's the experiment that we did with Asad. So we speed up the decrease in pH earlier during the development of the embryo to study the impact of acidification on that embryonic development. So that's the experiment. Hopefully you can see it in the screen, where we actually use citric acid at several different stages, day 13, day 15, day 17, or day 19. So we were trying to reach that final pH of 6.88 in the yolk and 7.18 in the albumen earlier in the embryonic development, hypothesizing that the earlier we acidify, the faster the mobilization of nutrients, and therefore the faster the embryonic development. And I'm going to show you one example, which is yolk. And when we acidified yolk at either day 13, 15, 17, or 19, we did see an increase in the body weight of the chick at hatch, a decrease in the residual yolk post-hatching. And as you see here, with these very significant results, the mobilization of the yolk increases. So these represent the yolk weight before or after acidifying the environment to the final pH of 6.88. 88. So in any step of the way, when we were acidifying it at day 13, we looked at the next day, 14. When we acidified at day 15, we looked at the next day, 16, and so on. And what you see is a much faster hydrolyzation of fats and proteins present in the yolk when we acidify the environment. And that has an impact on a larger chick at day 1. So this is, and I'm going to, because I'm running out of time, beautiful pictures that explain you how this all happens, really. And these pictures are about the yolk sac. So we all see the yellow yolk or orange yolk. And well, how is that happening? Well, the container of the yolk sac, is what it looks a little bit. Those of you who have seen micrographs of the small intestine, the villi, etc., it looks a bit like that. And in fact, the yolk sac, to a large extent, is the precursor of the small intestine in the post-hatch. That yolk sac will be reducing as the embryo develops, and at later stages, the yolk sac internalizes and it is actually functioning as the small intestine. So this is how the yolk is being absorbed, and those cells that are present in these villi of the yolk sac are the ones producing the enzymes, lipases, aminopeptidases, and so that will be, in fact, digesting the yolk, absorbing the yolk, the yolk, that is cartoon, transferring nutrients into the bloodstream, the circulatory system of the embryo. In other words, if we acidify the yolk to the left, the yolk sac membrane released enzymes will be more active because the atmosphere is actually more, the environment is acid, and therefore the activity of the proteases and lipases increases. There's more release of nutrients. They are being absorbed through the yolk sac into the bloodstream of the embryo, and the embryo is when the embryo increases the growth and reproduction. I'm going to take 3 or 4 more minutes to very quickly drive you through the other strategy that we have been working in our group, which is directly applying novel supplements, particularly essential oils, and how these essential oils may improve embryonic development with some particular interest in the immune system and the digestive system. Now, initially we tested a wide range of essential oils, and what we have is that a number of these essential oils will have a positive impact on the expression of genes that are related to, one, gut integrity and tight junctions. So some of these essential oils that we've tested, and I'm thinking of ylang-ylang, I'm thinking of lemon myrtle, clary sage, and so on, have a positive impact by improving the expression of these genes that are related to the gut integrity. Now, if I look at the correlation matrix between these 7 genes, you will see that most of them are highly correlated. In other words, those essential oils that are improving one of the indicators, one of the gene, in fact, are improving most of them because they are highly correlated. In a second experiment, we looked at the impact of these essential oils, and I say 22 in this case, on nutrient transporter and digestive enzymes in day 1 chick. And a similar story, some of these bergamot, black pepper, turmeric acid, ylang-ylang improve the expression of some of these nutrient transporters and digestive enzymes compatible with a better development of the digestive system. Again, those 6 genes that we tested were highly correlated. You improve one, you're improving all of them. That's what the correlation tells. And finally, of these essential oil stories about the impact on the immune system, which is the other system that's lagging behind at day 1 post-hatch. And those essential oils that I'm looking here, ylang-ylang again is quite persistent, star anise, spearmint, patchouli, etc., black pepper again, are improving what looks like improving the readiness, the activation, if you want, of the immune system early in the life of the chicken, and again, quite highly correlated amongst themselves. And I'm going to be finishing probably 4 minutes later than I expected, but just a summary. I know I've been quite ambitious, and I apologize because I'm talking about 4 different themes in one presentation. But I wanted to have a good view of what we've been doing. First, I've talked about maternal conditioning and how plant volatiles, they look like they play a fundamental role in those innate or immediate post-hatch preferences, selection of foods by the day-old chicken. We've looked at what looks evidence of strong epigenetic mechanisms that will influence the progeny through the nutrition of the broiler breeder. And then I've spent the last 20 minutes talking about the in novo interventions that we've gone through, such as controlling acidification, and impressive results, indeed, one of my favorite topics. And then the use of a number of different essential oils, which show fantastic potential to improve the readiness, the robustness of the day-old chicken. Time for acknowledgement. When we started this project 6 years ago, the help from Professor Sehava Uni and Jonathan Dayan from the Hebrew University of Jerusalem was fundamental. They are the big gurus in in novo treatments. Thank you to them. And to Professor Peter Furkett, another of the big gurus of in novo. It also would like to acknowledge the fact that Peter welcomed my student Assad, and he spent there 8 months learning a lot from him. Also, I would like to acknowledge, the main funders of this research, the AgriFutures Chicken-Meat Program, the collaboration with the Dawala Group. Thank you so much. Without you guys, it would have been much more difficult. And the steering committee of the project that most of the data comes from, Rod, Greg, Sheridan, and Sarika. Thank you all for your help. And of course, thank you to all my team, Nuts and group. Thank you. You're lovely, and I really appreciate your support. Mike, back to you. Sorry, I went a bit longer than I expected, but I think we still have a few minutes left.
Yes, indeed. Thank you so much, Eugeni. Indeed, we have time for questions. So I'm sure that this has raised a lot of questions in people's minds. So please write your questions in the Q&A, and we'll get to as many as we can before 1 o'clock. So start off with a couple that have already been put in there. So the first one relates to the reduced protein treatments that you talked about. And I guess it's a question around how you balance that. So the question was around dietary lysine content, whether you kept that constant or how you balanced basically high protein and reduced protein treatments.
Yes, that's a crucial aspect of when we play with different protein levels of different nutrients. The challenge is always balancing the rest of the nutrients. You want to be testing only one nutrient. So if many things change, then it's unlikely that you're going to find any conclusion. So good question, good point. I didn't show the feed formulations. I thought was not maybe the audience that I thought I was going to have, but it certainly has a lot of feed formulation exercise to keep the diets isoenergetic in terms of metabolizable energy, and we also keep the diets isoaminoacidic, so they have different levels of proteins, but we maintain the same level of the essential amino acids, particularly those who believe are limiting, and I'm thinking of lysine, methionine, threonine, etc. So they are isoaminoacidic, isoenergetic diets. when we compare those two different levels of crude protein. The downside of it, well, we have to play with different crude protein sources. Protein sources. We have to play with different amounts of crystal amino acids added to the diet. So it's inevitable, even if you have these isoenergies and isoamino acid diet, that there will be other differences in the feed formulation themselves. It's impossible to avoid that. I hope that answers the question.
Thank you. So a second question relates to the essential oil trial, so the in ovo injection of essential oils. So the question is, what's the control treatment? So presumably there was a control injection. What was in that?
Yes, thank you. There has been a lot of background work doing this. I did acknowledge some of my colleagues from Israel and from the United States who have helped us getting up to speed on the technique and the procedure. But we didn't shortcut. We did go through a lot of trial error stuff. So we tested everything and all potential control treatments that you can think of. So we've adopted finally. So one obvious control treatment is untouched eggs, so we don't inject nothing at all. The other more closer control, if you want, is injecting saline solution. Now, over the years, we've learned that we never find differences between the untouched egg, the non-injected, and the saline injected control. Never a difference, at least in terms of hatchability and the health of the day one chick. Over the years, we have then adopted as the main control the saline injection, because we believe that that's what allows us better to study, say, one essential oil. So I'm injecting saline solution, and I'm injecting ylang-ylang in a saline solution. So the only difference between the two injections will be basically the essential oil, and that's what we use as a control.
Okay, thank you. I have a question here. I'm not entirely sure what it means, but I guess it's asking for a comparison between what you've talked about and what happens in layers. So the question says, do you think this also could happen in layer breed? I guess it's how widespread do you think these observations could be beyond the 3 weeks you've been talking about?
Sure. So the answer is yes, of course it does. The physiology of the reproductive system will be essentially the same in broiler breeders and in laying hens. The only difference is that laying hens will not produce fertile eggs, of course, and they produce table eggs. Now, maybe, well, some of us or some of you may have experienced that if you take eggs from different farm environment XYZ with laying hens that might be just free range and might have access to different plants, and you can really taste difference. So I've tried that doing some sensory sessions, and you can taste the difference between different sets of eggs if the laying hens have been reared in very different environments, having access to different feed ingredients, basically. And that's due to the transfer of volatile compounds. So yes, the answer is, of course, it happens. So there's a number of volatile compounds in the feed and the feed ingredients that will be transferred to table eggs. The reality is that in the commercial production of table eggs, there's quite uniformity in the type of diets that we feed laying hens. Very well balanced diet, antibiotic free, etc., chemical free, but very, very well balanced. And all the laying hens will be eating very similar diets at the end of the day. So you will not, if you go to the supermarket, you will be rarely find differences in the sensory perception of the eggs, and that's because the volatiles that are being transferred will be very, very, very similar. But it does happen. We wouldn't talk about transgenerational or intergenerational because the eggs are not fertile, but the principle would certainly be the same.
Okay, I'm following up on that in terms of the volatile transfer. It's a question for me, this. So you've shown that you can transfer volatiles from feed through mother to yolk to the growing in embryo chick. I guess my question is that the feed volatile perception is through the equivalent of smelling, so it's through the air. Whereas when you're transferring it, it's all going through plasma and in a liquid phase, as it were. So are you saying then that transfer through plasma and liquid leads to the same final receptor mechanisms in the brain? Is that your hypothesis? That you're triggering that same response because the end destination point is the same, whether it's a placental delivery system or an aerosol, if you like, from the feed. Is that the assumption here?
Certainly so. Of course, there's much research that probably needs to be done in the future to close the circuit. That's certainly the hypothesis. But also, I would like to emphasize the concept that some of you may be familiar with, but the fact that these olfactory receptors, that happens also with taste receptors, are not only present in the olfactory mucosa. We know now that these olfactory receptors, you'll find them present in pretty much in many tissues all over the body of us or the chicken or the mice. Most of the research has been done in mice. And they activate the cell in different ways and with different functionalities. Now, if that olfactory receptor is present in the olfactory neurons of the olfactory mucosa, it will elicit smell. If that olfactory receptor is present, say, in an enterocyte or an enteroendocrine cell in the gut, we'll probably release a gut-brain axis response that has nothing to do with the smell. Same receptors, different cells, different responses, and different functions.
Okay, thank you. So one final question. It's looking to the future. So what are the potential long-term benefits of essential oil in ovo applications on post-hatch and performance of chickens? So do you think that this is going to be, or is, or could be a commercially applicable technology?
Excellent question. Thanks. Yes. We obviously, every bit of research we do is ultimately thinking of adoption by industry. Make no mistake. So we're doing, I haven't emphasized it at all. I just went maybe too quick, and I apologize for that. I wanted to explain so many things. But what we do with in ovo injections is always go through, or most of the times go to the day 17 of embryonic development, or the 18th day of embryonic development, and not before and not after. Why? Because of industry adoption. Industry, they already have a practice of in ovo injection for the Marek disease vaccine, and they do that during day 18 of embryonic development. So if we can develop functional practices that improve embryo development, and we can use it in the same window of the day 18 of embryonic development together with the Marek's disease vaccine, then we have a high chance of industry adoption. Industry would not be injecting twice, basically. So we're trying to adapt our strategy to the day 18 injection. And the answer is yes, we have done that. And truly, a number of biomarkers. I didn't talk about the immune system. One of the PhD students, Mila Meyer, who just finished last week, actually, submitted the thesis, did a wonderful job looking at the impact of carbapenem on the immune development in chickens. And the results are fantastic, and they are already published. So happy to contact with you, whoever is interested, and pass you through these publications. Thank you.
That's great. Thank you so much, Eugeni. Thank you once again for such a broad-ranging and excellent presentation, and for answering all the questions so clearly. If you could go to the next slide, please. Then it's just a reminder to those of you tuning in that this is what part of an ongoing sequence of Coffee Science seminars. The next science seminar should be in 2 weeks' time, so stay tuned using the web address or the QR code that you can see on the screen, and you'll learn more about the upcoming seminars. And in the meantime, thank you everyone for your attendance, and thank you once more, Eugeni, for today's seminar. Thank you and goodbye.
Thank you so much. Thanks, Mike. Thanks, everyone. Thank you. Bye.